Welcome back. We're still touring the cell. We're looking at cell structures. In this video, we'll look at the mitochondria, chloroplasts, and peroxisomes. So let's check these guys out. We are moving through a lot of slides at this point. Okay, so we want to take a look at mitochondria. Mitochondria are not a part of our endomembrane system, but they are bound by two membranes. So let's look at their structure and their function. And probably you learned that the mitochondria are the powerhouses of the cell. We are going to spend an entire chapter focusing on how they're the powerhouses of the cell. That's chapter 9. So we won't go into a lot of detail here about what they do, but structurally, let's check them out. Structurally, these organelles are made up of two membranes. The outer membrane is smooth, as you can see, and sometimes they're drawn like this, and the inner membrane is highly curved, so it has a much larger surface area than the outer membrane. And then there's a space. There's the matrix in the middle, okay, and then there's the space between the membranes, so we've got a couple of spaces where things can happen. Here are a few interesting features about the mitochondria. They have two membranes. They have their own DNA, and they can, with their own ribosomes, and they can make their own proteins. They also move around independently inside cells, and they can reproduce independently. There are somewhere around, there can be as few as one mitochondrion in a cell to thousands of mitochondria in a cell. Just depends on the cell type. Now, where they come from, they don't seem to emerge from the endomembrane system. But if you're interested in their evolutionary history, check out the Endomembrane Theory. Researchers believe that mitochondria and chloroplasts probably evolved as bacteria that had taken up a home inside a eukaryotic cell. The inner membrane of the mitochondria is a lot like the membrane of a bacterial cell. The DNA and the ribosomes in mitochondria look like DNA and ribosomes inside bacterial cells. And there's a lot of other evidence for this. But in any case, they have an important job to do. This is the site where cellular respiration happens. This is the site where cells can take nutrients and use those nutrients to make an abundance of ATP. ATP is a molecule that does cellular work. It's kind of the currency of our cells, right, to do work. So mitochondria don't create energy, but they transform it. They take nutrients, like glucose, and use the energy in those nutrients to make ATP. And that happens here in the mitochondria. If your mitochondria don't work properly, maybe there's a mutation in some of the mitochondrial DNA, and you produce a gene or a protein that doesn't work quite right, you might not make enough ATP to power your cell. And you may have symptoms in parts of your body that need a lot of ATP, like your muscles or your visual system. So diseases of mitochondria tend to lead to less functional mitochondria, and that can be really a problem. Here's a response question. I want you to check it out for our extra credit assignment. In a fungus called Podospora anserine, researchers have shown that as the fungus gets older, the inner membrane of its mitochondria becomes less and less folded. What do you think happens to the function of the mitochondria as it ages? The question really is, why is this inner membrane folded? Why is that useful for the mitochondria? And what happens when it becomes less folded? Check it out. Chloroplasts are another organelle that we see, but we see chloroplasts in plants. Okay? Chloroplasts are a type of plastid. Plastids are double membraned structures that are found in plants only. We think that they probably arose in a way similar as mitochondria did, that they arose as bacteria living, as algae living inside a bigger plant cell. But check that out if you're interested. In any case, every plant, every chloroplast has a double membrane. Okay? This time the membranes are both smooth, and inside that double membrane are stacks of little membranes called grana or granum. And these stacks, the membranes themselves are called thylakoid sacs, and then there's this fluid-like structure called the stroma surrounding it. We're going to check out what chloroplasts do in Chapter 10. That's the focus of Chapter 10, but we want to at least write down a couple of things. They're found in plants. Their structure is a double membrane structure with thylakoid sacs inside. These thylakoid sacs contain pigment molecules that can absorb light energy, all right? And they also have their own DNA. They have their own ribosomes. They can make their own proteins. They reproduce independently inside plant cells as well. Their job, of course, is the site of photosynthesis. This is where plants can take energy from the sun, absorb it somehow, and use it to make sugars. They also need to grab carbon dioxide as a carbon source to build their sugars. So they can grab carbon dioxide using energy from the sun, make sugar. It's pretty amazing. That's photosynthesis. We'll check that out later. And then finally is the peroxisome. One last structure to take a look at. Peroxisomes are tiny membrane-bound structures. They're about 1 micrometer in size that are filled with enzymes. They're filled with what are called oxidative enzymes. So tiny membrane-bound structures filled with oxidative enzymes. And those are oxidative enzymes. Oxidative enzymes are enzymes that catalyze oxidation reduction reactions. But in any case, their main job, well, they have 2 jobs. One of the things that they can do is that they can detoxify poisons. And they detoxify poisons by adding or transferring hydrogens to oxygen. Right? So basically, they can take poisonous molecules and make them less poisonous. That's good. The second main function of the peroxisome is to break down fatty acids from fats into small pieces so that those small pieces can be used by mitochondria to make ATP. And so we often find peroxisomes sitting very close to mitochondria because they help break down these complex fats into small pieces that can be used to help make ATP or to help with cellular respiration as the nutrition for cellular respiration. That's it. I think that's the tour of the cell in terms of structures in the cell.